Okay, so hi everyone. I'm Jay Sung, and I'm an assistant professor at Mayo Clinic in Rochester, Minnesota, where I run a computational research group focusing on advancing systems medicine for autoimmune diseases like rheumatoid arthritis. Today, I'll be talking about a paper that came out in mSystems in November 2022 on how we developed a bioinformatics tool to identify biosynthetic gene clusters of known secondary metabolites in microbial shotgun metagenomes. Here's the paper, which was led by Dr. Vinod Gupta and Utpal Bakshi, who are postdoctoral fellows in our group while performing this work. So, what are secondary metabolites? And also called natural products. These are bioactive and structurally diverse small molecule metabolites from secondary metabolism, which are metabolic processes not involved in essential functions like growth, development, and reproduction. And the reason that microbes make secondary metabolites in the first place is so that they can interact with other microbes or with the environment. A couple of examples include the bacteriocins that microbes use to kill off competitors, or siderophores, which are mainly iron chelating molecules, as many people know, but also have a role in shaping community dynamics. But do secondary metabolites affect human health? Well, absolutely. Traditionally, these secondary metabolites have been an important source of a variety of medicines. So the translational impact of understanding how, when, and why microbes make secondary metabolites is going to be quite significant. But how do microbes make secondary metabolites? It's by using biosynthetic gene clusters, or BGCs, which are physically clustered groups of genes in a microbial genome that encode a biosynthetic pathway for a secondary metabolite and its chemical variants. As you can see in this example, the cluster of genes here encode for mega enzymes. So each of these codes for mega enzymes. And what I mean by mega enzymes is enzymes that each catalyze multiple reactions. So this small molecule here is taken across this assembly line of chemical transformations to make disorazole A1. We found all this to be very interesting as it explains the basis of chemically mediated interactions within a microbial community. What was really interesting to us is that BGCs have been discovered to exist quite prevalently in the human microbiome. This seminal work shown here by Mohammed Donia and Michael Fischbach a few years ago was able to predict putative BGCs in these taxonomic families here. And we're even able to detect BGCs made by these isolates here from the human microbiome, from HMP metagenomic samples. Now, inspired by this work, we asked the question of, in our own metagenomes of interest, say from fecal samples from patients with an autoimmune disease, can we identify experimentally characterized BGCs to help us better understand what may be functionally going on? And on a more translational side, can looking into BGCs help us find microbial metabolites that our microbiome spits out into the microenvironment that may be disease initiating, disease mediating, or even disease protective? So all we needed were the right tools to answer these broad questions. Well, although there are currently existing tools out there, and maybe you've heard of them, we found several shortcomings after using them ourselves. For example, most of these are for predicting unknown putative BGCs, where we don't know the final product, but we're only interested in the ones whose bioactive metabolic products are already known. They also provide predictions on a limited number of BGC classes, but we want to screen for all known classes. Some require a lot of fine-tuning, and none identifies the microbial source of the BGC. But we also want to know which microbes harbor the predicted BGCs. So to be fair, this is all probably by design, and hence more of a feature than a bug. Still, these are shortcomings based on what we wanted to do. But when testing these tools on our own simulated metagenomes of known community composition, we found that these methods leads to a ton of false positives. Hence, these shortcomings really limit their use for a wide range of metagenomic datasets. Thus, our goals were threefold: to develop a new computational tool that addressed the limitations of current pipelines, benchmark our tool on simulated metagenomes of mock microbial communities, and identify BGCs and their known secondary metabolites in different human body sites and diseases. So first, we made TaxiBGC, which stands for Taxonomy-Guided Identification of Biosynthetic Gene Clusters, which is a command line tool for predicting experimentally characterized BGCs and inferring their known SMs in metagenomes by first pinpointing the microbial species likely to harbor those BGCs. So the intuition of TaxiBGC works like this. So with a microbiome sample of metagenome reads in step 1, TaxiBGC finds which microbial species are there with the assist of a widely used taxonomy profiling tool called Metaflan. And then in step 2, TaxiBGC uses a predefined reference database to find which one of these species have BGCs, and thereby defining the search space of all possible BGCs that could be in your metagenome. And then in step 3.1, TaxiBGC does read mapping on the BGC genes in the metagenome to confirm in silico that the predicted BGCs are indeed there. For this read mapping step, we use 2 thresholds that eventually determine the presence of a BGC gene and the presence of an entire BGC. I'll talk about this a bit later, but these thresholds were determined using by benchmarking on simulated metagenomes of known community composition. At the end of this step, we get a final list of BGCs. And then in step 3.2, TaxiBGC provides the secondary metabolites known to be produced by those confirmed BGCs. Hence, the input is a shotgun metagenome, and what comes out is a list of BGCs and the SMs made by those BGCs. Now, the list of taxonomies that are known to harbor BGCs in their genomes, as annotated by the MyBig database, is shown here. As you can see, our TaxiBGC reference database takes into account a wide range of taxonomies when calling the potential list of BGCs from taxonomies found to be present in the input metagenome. We also provide a detailed characterization of the relationships among microbial species, BGCs, and SMs in our reference database. Very briefly, we take into consideration 390 total species after finding these in common between MyBig and the Metaflan 3 marker database. Hence, in the current version of TaxiBGC, the presence of up to this many can be queried in a metagenome. Now, among those 390 species, we show how many BGCs each species harbors, and many more than others. As you can see, we show the distribution of gene count composing the BGCs, and as you can see, there's a wide variance with the BGC encoding for colicin V or colicin 5 has 164 total genes, and these numbers of BGCs belongs to these BGC classes, with the non-ribosomal peptides or NRPs and polyketides with the largest sum of BGCs, while alkaloids here have the fewest number of BGCs. And finally, we show the BGCs rank ordered by how many secondary metabolites they make in descending order. These two BGCs can make up 13, these two BGCs here can make up 13 metabolites, while the majority of BGCs, as you can see, make only one secondary metabolite. Now, of course, building a new tool, we need to benchmark it against simulated metagenomes, where we know all the answers beforehand. We first need to do some parameter tuning, then see what the prediction algorithm is robust against, and then test prediction accuracy. So I skipped over this when talking about TaxiBGC in the earlier slides, but our method does use 2 crucial thresholds: the minimum BGC gene presence threshold, or for the minimum BGC presence cutoff, that is percentage length of a BGC gene covered by a metagenome reads, and the BGC coverage cutoff, which is the percentage of total number of genes present for a particular BGC. BGCs that pass these 2 criteria were considered present in the given metagenome, but how did we determine these thresholds? Well, in figure 3A and B of the paper, we show the pair of optimal thresholds. that give the best predictive performance in TaxiBGC. In addition, we found that the performance in predicting BGCs is robust against sequencing library size of the metagenome, and also against the relative abundance of BGC harboring species. We next compared TaxiBGC, which first identifies microbial species, versus kind of like the current standard approach of predicting BGCs by just mapping reads onto known BGCs. As you can see here, based on F1 scores, and which I know the equation here looks kind of funny, but it's just the harmonic mean of sensitivity and specificity, and also on the positive predictive values. You see that our taxonomy-guided approach predicts BGCs more accurately than by directly detecting BGC genes. We looked into why this is so, and it was mainly because TaxiBGC was able to keep false positives relatively low, and thereby addressing, I would say, the main limitation, or one of the main limitations of current approaches. But to be fair, we also need to show for both methods the sensitivities, or how accurately the actual present BGCs are predicted to be in there. In this case, then the approach that looks for BGC genes, or direct gene mapping approach, is clearly the favorite. But again, this comes with the cost of high false positive rate, favoring our approach to be the more conservative. Having developed TaxiBGC, we next wanted to see what BGCs and their SMs can be characterizing different parts of the human body. So we ran TaxiBGC on HMP samples, and this happened. In this heat map, I'm showing the prevalence of BGCs and metagenomes sampled from 5 human body sites. Columns are for different body sites: the vagina, mouth, guts through stool, nose and skin through the back of the ears, and the number of samples we're working with in the parentheses here. Let's see, on the rows, we're showing the BGCs found in the body sites with their MyBig database IDs and known secondary metabolic product. Colors here represent whether the BGC was found exclusively in one body site or multiple body sites. Colors here represent the BGC class. Colors on the heat map show the proportion of body site samples the BGC is in. And the stars that you see here indicate whether the BGC is significantly overrepresented in one or more body site. So now zooming out a bit, you can see that different body sites have different BGCs at different proportions, with some a lot higher than others. Like BGCs for gastrin are found exclusively in the vagina, and BGCs for these mucosins here and fatty acids are found to be exclusively in the oral cavities. Now, I want to bring attention to these BGCs here marked in orange. And these BGCs encode for bacteriocins, or a group of antimicrobials. And hence, we see that BGCs for bacteriocins are prevalent in microbiomes throughout the human body, confirming what other researchers have previously seen, that microbes of any complex community basically make a ton of antibiotics depending on nutrient or space availability, and that these antibiotics have a lot to do with governing ecological stability. Next, we look into whether gut microbial BGCs have any association with human diseases, given that their products could be bioactive inside their host and doing something to our immune system. So we ran TaxiBGC on public data sets for liver cirrhosis, rheumatoid arthritis, Crohn's disease, and colon cancer. Purple bars will indicate the prevalence of that BGC in healthy controls. Yellow bars are the prevalence in cases. So prevalence you can think of as the frequency of presence and absence of those BGCs. So if all the samples have a certain BGC, then that's 100%. If none of them have a BGC, that's 0%. Okay, so assuming you get the picture. As you can see, some BGCs are found more frequently in disease, such as the ones here, 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 here. Okay. And some are more are found more frequently in controls, such as ones here, here, and here. You can notice that some signals are consistently found in more than one study, such as for niacin O here, and ruminococcin A here, gastricin T here. Now you may notice that some BDCs are found in common for multiple diseases, like niacin O here, and gastrin S. Okay. And precisely, we report for the first time that some gut microbial BGC associations are shared among multiple pathologies. What's interesting is that these BGCs are found in the gut microbiomes of diseases of the gastrointestinal tract, perhaps hinting that from a microbiome perspective, these GI diseases have more in common than we thought. So for those who want to use TaxiBGC, we have a GitHub page to go along with our paper. This repo, along with the conda package I'll show later, was designed by a very talented and super sharp undergraduate in our group, Daniel Cheng. The GitHub provides a friendly guide on how to install and use TaxiBGC. We also have a demo. You put this in the command line here, and TaxiBGC gives you these output files. And for this sample, you get a list of BGCs. So for this sample here, you get a list of BGCs, the secondary metabolites they encode for, the classes of those secondary metabolites, and the source organism of those BGCs as annotated in the MyBig database. We also have a conda package, so you don't have to download all the software dependencies required by TaxiBGC. The single command line here, it'll download everything for you. Okay, so I'll wrap up. We developed TaxiBGC, a command line tool for predicting experimentally characterized BGCs in metagenomes. And here, experimentally characterized means that we know the metabolic product. And I wasn't able to get into this in much detail, but actual users of our tool will soon notice that TaxiBGC provides a significant advantage over currently existing methods. We were able to associate BGCs and their SMs with different human body sites and with multiple GI diseases. And this perhaps provides a potential starting point for discovering disease biomarkers or even therapeutic agents from the gut microbiome. And ultimately, we expect our tool to help future studies into the chemical ecology of diverse microbiomes. So if you have metagenomic data, be it from the human body, model organisms, or the environment, soil or ocean, and you want to characterize what BGCs may be in there to probe what secondary metabolites could be mediating microbe or microbe-host interactions, then please try out our tool or send me an email and perhaps we can collaborate. Thank you for listening, and I hope everyone has a wonderful day.